Good morning, and this is today we are starting the second topic of chemotherapeutic agents. And in our past topic, we saw the different type of antibiotics and how they are effective on different type of microorganisms. So today we'll start with the broad spectrum antibiotics. So broad spectrum antibiotic, as we know, these type of antibiotics is effective on the multiple type of microorganism. So first one is the tetracycline and chloramphenicol is there, and they are broad spectrum type of antibiotics. And these type of antibiotics still we are using, and we have multiple type of first generation to fifth generation antibiotics, and they are called broad spectrum because their effectiveness against wide range of microorganisms like gram positive bacteria and gram negative bacteria. So gram positive and gram negative bacteria both are having sensitivity on this tetracycline and chloramphenicol, and they are not resistant. And some few organisms only they are tetracycline resistant, chloramphenicol resistant, but most of the bacteria they are sensitive on the tetracycline and chloramphenicol. So rickettsia and pneumonia, chlamydia, anaerobes, and sirocates and pyroli, and some protozoolite, malarial parasite, and ant amoeba. They are all sensitive uh, on the tetracycline and chloramphenicol. So first we will start with the tetracycline. Tetracycline is a first type of broad spectrum bacteriostatic antibiotics, and the how it will function. It is a type of bacteriostate. Means it will not increase the when we are giving these tetracycline to patient, the bacterial infection, the count will not increase. So it is a type of bacteriostatic antibiotic. So it is active against the many gram positive and gram negative type of bacteria, including anaerobes, rickettsia, chlamydia, mycoplasma, protozoa, or especially and amoeba. So if you'll see the mechanism of action, it will inhibit the bacterial protein synthesis by binding and interfering with the ribosome. So how it will interfere in the mechanism of microbes? It will bind with the ribosome and it will inhibit the translation of the protein. So when the mRNA is binding to ribosome and tRNA transfer the amino acid to ribosome, and the protein will synthesize. So what it will do, this tetracycline, it will go and bind to ribosome. So the protein can't able to synthesize on the ribosomes. So it will inhibit the mechanism of translation basically. Now uses and indication. So look, chlamydial infection, including sexually transmitted disease, in combination with aminoglycosides indicated for plague and tularemia. So these are all diseases are having sensitivity on the tetracycline, and still we are using these tetracycline as a effective antibiotic. So many type of sexually transmitted diseases there, and most of the important disease is chlamydial infection and gonorrheal infection. And these infection we can reduce or from the uses of the tetracycline, and many other very important diseases like tularemia, plague, and brucellosis is there, and they are all treated with the tetracycline antibiotic. Now, if you'll see the classification, there are three type of we are dividing tetracycline, and short-acting tetracycline is there, long-acting tetracycline, and intermediate tetracycline is there. These classification is depend on how their time, on the duration of the timing where they are effective. So short duration means 6 to 8 hours only they are effective, and after that they will start degrading, and then we have to give fresh dose. So these type of short acting antibiotics will work only 6 to 8 hours. So chlorate tetracycline is there, then tetracycline and oxytetracycline is in the short acting antibiotics. And long acting antibiotics also is there, and they are effective on 16 to 18 hours. So the example is D and tetracycline. So these two are the not in long and not short, but intermediate acting antibiotics. Then intermediate acting antibiotics there which will be functional up to the 12 hours. So doxycycline and minocycline is the example of long acting antibiotic. Then contraindication and precautions. So when we are giving the tetracycline to patient, it will cause the hypersensitivity, and precaution always we have to take when the patient is having renal disease and hepatic disease history. So those patients which is having hypersensitivity issues and renal disease and hepatic disease history, we should not give these tetracycline. And adverse effect, if we will give the high dose, so mild nausea, anorexia, bulky and loose stool, hepatotoxicity, flatulence, and pancreatitis, it will be there. And these are all the side effects. It will be there when the continuously we are treating with the tetracycline and the higher dose if we will give. Now, other drug interaction, if you will see, milk and dairy product, magnesium containing laxative, antacid, calcium supplement, iron supplement, reduce absorption of tetracycline. So when we are giving the tetracycline to some patients, we should not suggest that they have to intake the milk and dairy product, and magnesium containing the laxative agents, and antacidic like calcium supplement also, or iron supplement. These supplements we have to avoid when we are taking the tetracycline. Now, when we are giving with the combination of penicillin and tetracycline, what tetracycline will do, it will decrease the effectiveness of penicillin, and hormonal contraceptive drugs also it will decrease. So always we have to avoid the tetracycline in the combination of penicillin and hormonal contraceptive drugs, because what tetracycline it will do, it will decrease the effectiveness of penicillin and contraceptive drugs. Then nursing responsibility, what we have to see, they should not be used during the pregnancy and lactation and childhood period, because it is having hypersensitivity and hepatotoxicity and renal failure effect. That's why we always have to avoid the in the pregnancy, lactation, and childhood period these tetracycline type of antibiotics. Then they should not be given when renal functions are impaired, and the patient which is having renal function dysfunction history. So those patients we should not give this tetracycline. Then instruct the patient not to take antacid, calcium supplement, iron supplement, and magnesium containing laxatives, and do not take milk or dairy product within 2 to 3 hours taking tetracycline, because when the patient is taking these are all as a food intake, the tetracycline effect will be changed, and then it will react and it will create side effects. Next is chloramphenicol. So chloramphenicol is a broad spectrum type of antibiotic, and it is effective against on the aerobic, anaerobic, gram positive, gram negative, and rickettsia, and they are closely resemble in the action of the tetracycline. That's why we are studying tetracycline and chloramphenicol together. So it is also a bacteriostatic like tetracycline, and Haemophilus influenza, Neisseria, meningitis, and they they have particular effect like bacteriostates. So the chloramphenicol, what it will do, it will not allow the bacteria to grow and develop. Just it will control their population. So slowly, bacteria population will be decreased, and it will slowly remove the infection. So how will these chloramphenicol will work? So it will inhibit the protein synthesis in the susceptible bacteria, and in the presence of these chloramphenicol, organism cannot multiply, and it acts as a bacteriostatic drug. So as we saw that how the tetracycline is functioning on the protein synthesis, and same type chloramphenicol also will function against the protein synthesis in the bacteria. It will same type, it will bind with the ribosome, and then it will inhibit the protein synthesis or translation. Then indication and uses. So this chloramphenicol type of antibiotic mainly used in the typhoid fever, urinary tract infection, rickettsial, chlorella, and bacterial meningitis, eye, ear, and these type of infection. So mainly it is basically use of the eye, ear type of and urinary tract infection here utilized. Contradiction and precautions will see history of hypersensitivity, toxic reaction, pregnancy and lactation, precaution for renal impaired and hepatic patient. We should not give these chloramphenicol and tetracycline. So if you'll see the reaction, effectiveness, mechanism, and precaution and side effects. These are all same with the tetracycline and chloramphenicol. That's why we are categorized together, and their effect also is same, like they are doing nausea, vomiting, diarrhea, oral vaginal and candidiasis, bone marrow depression, hypersensitivity reaction. So these are all different type of side effects there, and especially the patient in combination when we are treating with the chloramphenicol and as well as tetracycline, these side effects will come. Now other drug interactions, if you will see the tetracycline inhibit the mechanism metabolism of tobulidamide and chloramphenicol, warfarin, cyclophosphamide, and phenylethylamine. So when in combination with these are all antibiotic, when we will get it will inhibit the chemical metabolism. So chemical our metabolism it will affected by these tetracycline, and these are all type of antibiotic. So as the bacterial state action of the chloramphenicol can antagonize the cidal action of beta-lactam aminoglycoside on certain bacteria. So on particular some bacteria they will be having a negative effect when we are giving these chloramphenicol with the combination of beta-lactam type of antibiotic and aminoglycosides type of antibiotic. Now sulfonamide. Sulfonamides is another type of antibiotic, and it was discovered by the Gerard Domin in 1870s. So sulfonamides are the one of the oldest type of antimicrobial agent, and these derivatives of the parent compound para-aminobenzene and sulfonamide. So they are the mainly bacteriostatics, but at very high concentration they may have bactericidal effect. Like how the erythromycin in low concentration they are bacteriostate effect, and then we are increasing the concentration of erythromycin, so it will turn into the bactericide. Same sulfonamide also will do same type of effect when we are giving in the low concentration. It is bacteriostate. The population of the bacteria it will not allow to increase, and when we are giving very high concentration, so it will work as a bactericidal. Okay, so some example is there in the group: sulfadazine, sulfamethoxazole, sulfasalazine, cotrimoxazole is there. So these are all the member of these sulfonamide. Now mechanism of action, if you see, we already said they are bacteriostates, means they will inhibit the enzyme folic acid synthase, so folic acid is not synthesized, and when folic acid is not synthesized, and we know that folic acid is very essential for the bacteria growth, so that's why the bacterial growth will not be there, and it will work as a bacteriostatics. Okay, so the folic acid it will not synthesize because the enzyme which is required in the folic acid synthesis, it will not allow to synthesize the folic acid, so that the bacterial growth will be inhibited. Now use. So urinary tract infection or UTI, and ulcerative colitis, trachoma, conjunctivitis. So sulfonamide types of drugs we can utilize for the urinary tract infection or UTI, ulcerative colitis, trachoma, conjunctivitis, and acute bacillary and dysentry and canceroid and meningitis. Meningitis is a disease of meningeal layers in our brain, which is surrounded by the our brain. So infection in the meningeal membranes will be a very lethal type of effect on the neurological health. Now, contradiction and precautions. So, children younger than 2 years and pregnant and breastfeeding mother, renal and hepatic disease, hypersensitivity to sulfonamide drugs we should not give. So, again, the mother which is pregnant and breastfeeding mother, so we should not give to them, and the small children like less than 2 years, we should not give. And those patients which is having renal, hepatic, and hypersensitivity history, we should not give these sulfonamide drugs. Now, what type of side effect it will produce? So, fever, rash, blood, uh, dyscrasis, nausea, vomiting, aplastic anemia. These are all the side effect of the sulfonamide. Now, how these sulfonamides is interacting with other type of drugs? So, sulfonamides can increasing the blood thinning effect of the warfarin, possibly leading to abnormal bleeding. So, when we are giving in combination sulfonamide and warfarin, these two type of antibiotic can be have increasing effect of the blood thinning, and that's why we in combination of warfarin and sulfonamide we are not giving to patient. Then increases blood level of potassium may occur when sulfamethoxazole trimethopyrin is combined with the ACE inhibitor. So always we have to avoid the ACE type of inhibitor with the sulfonamide. So it will increase the blood pressure in the patient. So always avoid these type of combination. Then sulfonide may increase the effectiveness of oral hypoglycemic drugs, and may increase the effectiveness of oral contraceptive drug. We already know when we are giving with the tetracycline and other type of antibiotic in combination with sulfonamide, if two antibiotics have same type of effect, then the proportion will always increase. See, if antibiotic A is having effective and it is causing the side effect on the blood pressure, if it is increasing, if we are giving another type of antibiotic which is having same type of side effect, so both type will combine and their effect will be more. So that's why always we have to avoid those type of antibiotic which is having same type of side effect. Then quinones. Quinones is another type of compound which is functioning as an antibacterial. So antiamoebic compound also there, and these compound is against the amebia and amebiasis, and the amebiasis is caused by the, so it is a protozoal disease caused by the Entamoeba histolytica, and amoeba affect the liver, lung, brain, or other tissues. So primarily it will be effective on the intestine, and then later on in the secondary infection it will affected nearby the secondary type of organ like liver, kidney, and then it will travel to other type of tissues. So it produce the hepatitis and amebic disease. The drug used to treat this kind of infection we are calling anti-amebic drugs. So anti-amebic drugs is utilized not only particularly amebiasis, but other type of protozoan disease also we can able to control using these anti-amebic type of drugs. So amebiasis, if you see amebiasis, it is also called the amebic dysentry. So when Entamoeba histolytica infect any patient, so he will be high level of dysentery it will be there. So it is an infection of the intestinal tract when the uncooked material or contaminated water when we drink, and these contaminated food and water is having amoeba and amoeba cyst or spores or infection is, if it is there in the water or food, then we have the intestinal infection of Entamoeba, and the patient which is having infection, it will cause the amebic dysentery. So intestinal tract is the primary infection site, and the main causative agent is Entamoeba histolytica. So the disease can be acute or chronic. So with the patient showing varying degree of illness from no symptom to mild diarrhea to accumulating dysentery, and if the Antiamoeba infection is spreading nearby your organ, so these symptoms will also increase. So according to various degree, we have to treat it. So diagnosis established by the isolating Entamoeba histolytica from fresh fecus. So what we have to do is free fecus sample we have to analyze in the microscope, and we can easily see the Entamoeba histolytica while analyzing using the microscope. And when we see the Entamoeba histolytica, so we can start the antiamoebic drugs. So therapy is aimed not only at the acute patient but also at those who are asymptomatic carrier, because dormant Entamoeba histolytica may cause future infection in the and it will work as a carrier. So it is a potential source of the infection for others. So always we have to give the long duration of the anti-amebic type of drug, so that if the patient is carrier type, then he will not spread the infection to others. So it will kill the complete Antiamoeba histolytica in the. Then protozoal infection are commonly among the people in underdeveloped tropical and subtropical countries, and where the sanitary condition, hygienic practice, and control of vector of transmission are inadequate. So when we already know that the Antiamoeba histolytica infection is happened because of the people are drinking contaminated water and contaminated food they are taking. So through contaminated food and water, these Antiamoeba or many other protozoal infection are spreading. So always when we are using the water, we always have to boil it completely. Then only we have to use it. If we are eating the uncooked material, and these uncooked material is having Antiamoeba infection, then easily we can have amebic disease in our. Then mechanism of action of these anti-amebic type of drugs. So it requires reproductive activation of nitro group by susceptible organism. Then it really selectively toxicity towards the anaerobic and microaerophilic pathogens such as Entamoeba histolytica and the lambia, etc. These type of disease they are causing by the protozoa, and these disease is require the anti-amebic type of drugs. And these organisms contain electron transport components such as pyridoxine, small iron sulfur protein that have sufficiently negative redox potential to donate electron to metronidazole. So the most effective type of anti-amebic is metronidazole, and then we are giving this metronidazole to affected patient. So these metronidazole will be function as a electron transport component such as pyridoxine, small iron sulfur protein, and these parts will be affected by the metronidazole of the protozoa, so that they will be killed easily by the these type of compound. So single electron transfer form a highly reactive nitro-radical anion that kills susceptible organism by radical mediated mechanism that target DNA, resulting in cell death. So when we are giving any anti-amebic type of drug, especially metronidazole, it will target the DNA, and it will inhibit the various degree level of the DNA synthesis, transcription, so that on the various level it will be lethal for the bacteria and protozoa, and the resulting the microorganism will start dying. The single electron transport form a highly reactive nitro-radical anion that kills susceptible organism by radical mediated mechanism that target DNA, resulting in the cell death. So how it is happening? When we are treating with the metronidazole, this is the chemical structure of the metronidazole. Metronidazole is coming inside the bacterium. So metronidazole, what it will do, it will metabolize. It will be effective on the metabolites, and these metabolites inhibit the DNA synthesis and transcription, and DNA will be fragmented, and then their DNA will then it will be fragmented. So ultimately, cell will die, and so infection we can able to easily control. Here the one mechanism is involving when these metronidazole is inhibiting it is converting ferredoxin to ferredoxin, and it will reduce the ferredoxin to ferredoxin oxidase forms. So luminal amoebiasis, systemic amoebiasis, and mixed amoebiasis. These three type of amebic disease we are using these metronidazole type of anti-amebic drugs and act on a parasite in the lumen of the boil. That's why we are going to renal amebiasis is the against moving in the intestine wall and liver, and that's why we are calling systemic type of amoebiasis. And mixed amoebiasis are both in the human and systemic form of disease. It will be effective. So three type of infection it will be there when the infection on the human of the wall, then the luminal amoebiasis is there. If the infection is the amoebis in the intestinal wall and the liver, then systemic amoebiasis. And when the infection is the luminal and systemic form of disease, then it will be in the mixed type. So we have to treat in the both type of disease. Now classification, if you will see amoebicidal drugs. So according to site where the drugs is effective, the amoebicidal drugs are classified into 3 points. We already discussed these three sites like luminal, systemic, and mixed. Means mixed between the luminal and systemic two types. So these two, according to infection, we have to treat, and these three type of drugs we have to give. And first we have to see where the infection is there, and then only we have to decide what type of drugs we can able to give. So metronidazole are contraindicated in neurological disorder and blood discrepancies it will do. So that's why we always have to precaution when the patient is having neurological disorder and blood discrepancies. When cautiously used in the pregnancy and alcoholism, and those have hepatic disorder. Now side effects. So metronidazole it will cause the side effect of anorexia, headache, abdominal cramps, ECG changes, skin rashes, vertigo, nausea, vomiting, thrombophlebitis. These are all side effects it will cause. Now, how these metronidazole interact with the other type of drugs. So metronidazole, when we are mixing with the alcohol containing type of drugs, it will react with that, and it will produce the tachycardia, flushing, sweating, and vomiting. So, and concurrent use with the warfarin may prolong the bleeding, and thrombophlebitis may develop if IV infusion is not done with the dilution of sterile water. So these are all things it will be there, and we always avoid these type of combination of with the metronidazole. Now anti-malarial. Anti-malarial are those drugs which will be effective against the malarial vector, and these type of drugs are used for prophylaxis, treatment, and prevention of malaria. So they are act against the plasmodium, and it is a protozoa parasite and main reason of the malaria, and they are the using vector and mosquito, and through mosquito only they are transferring. So malaria is transferred by the biting of female anopheles mosquito. Another type of disease also there which is transferring by the mosquito biting. But if you see the plasmodium type of transferring, the mosquito female mosquito, when it will bite, it will transfer. So plasmodium, it is basically a protozoa and living inside the infected people. And when a healthy mosquito, female mosquito, will bite the and suck the blood, so blood is containing these protozoa spores, and it will transfer into the healthy mosquito. Then again it will start the cycle, and it will transfer these protozoa to healthy person, and it will cause the malaria. Now, how these anti-malarial drug it will function? So it will inhibit the protein synthesis and affecting the DNA RNA function. So anti-malarial drug when we will give, so it will stop the DNA and RNA function in the parasites, and these anti-malarial drug will stop the synthesis or inhibiting the protein. And how we can use an indication? So clinical cure and prophylaxis of all kind of malaria, we can give these anti-malarial type of drugs. So if you see the various type of anti-malarial type of drugs is available, and the basic type of anti-malarial is chloroquine, which is isolated from a plant, and primaquine is there, mefloquine is there, trimethylamine is there, propioniol, and artemether is there. So these are all different type of drugs is there, and these are all inhibiting the growth and development of the trypanosome. Then contradiction and precaution. So when we are treating the patient with these anti-malarial type of drug, it will cause the hypersensitivity, retinal damage, concurrent hepatotoxic drugs, pregnancy and lactation, renal insufficiency, known or suspected resistance, and P. falciparum infection. So these specific type of parasites, if we will treat with the anti-malarial type of drugs, so these type of side effect it will be there, including nausea, vomiting, seizure, headache, abdominal cramps, hemolysis, ototoxicity, and gastrointestinal upset, visual disturbance like loss of accommodation, blurred vision. So these type of side effect it will be there, and always we have to hypersensitivity and some type of hepatotoxic effect or renal hepatotoxic type of history, if it will be there with the patient, then always we have to avoid it. So the pregnancy and in the lactation time, these type of anti-malarial drug we should not prescribe. And drug interaction, if you see concurrent use of such compound with anti-antiamoebic, tricyclic, antidepressant, it may increase the risk of cardiac conduction of defects. So always we have to avoid these type of compound with the combination of antiarrhythmic and tricyclic antidepressant type of drugs. Then it will be interact and it will be uh, very adverse effect because in these when we are giving in the combination of these type of drugs. Next is anti-helminthic type of drugs, and these drugs is effective on the worm infection or particularly helminthic type of infection. And these drugs is useful for the specific type of infection. And it is very important to identify the causative worms, then only we can give these anti-helminthic type of drugs. So finding the parasite of the ova, larva, and fecal urine in the fecal urine, blood, and sputum or other type of tissues by microscopic identification or other latest technique like PCR and ELISA we can utilize, but we have to first identify the parasitic infection, then only we have to treat with the anti-helminthic type of drugs. Now, classification, if you will see the anti-helminthic type of drugs, so we are classifying on the basis of the effectiveness. So the drugs which is effective on the nematodes, we are classifying the nematodes, anti-nematodes type of drugs. So albendazole, mebendazole, pyrantel, panoate, fluconazole, piperazine, ivermectin, diethyl carbamazine, and thiamidazole, doxycycline. These type of drugs is effective on the nematodes infection. Then other type of drugs is there which is effective on the nematodes and metryptomate, oxaminiquine, triclebendazole. So these are all drugs is having effective against the nematodes infection. Next type of drug is the nicotinamide, and nicotinamide is effective on the gastro. Then praziquantel is there. Praziquantel is effective on the nematodes and gastros. If the patient is having both type of infection like trematodes and gastros, they have to give this praziquantel. Now, if you will see the particular worms or helminthes, and these worms is having particular type of combination we have to give. If the patient is having infection of tapeworm, so we have to give the nicosamide or paracetamol or albendazole. They will be effective on the one type of infection. Then around one type of infection it will be there. So if the Antirobabeus vermicularis pinworm infection is there, then an effective drugs it will be albendazole or pyrantel. Then Ascaris infection is there, then albendazole or pyrantel, then albendazole or albendazole we can give for the whipworm. Albendazole and thiabendazole is effective on the trichinosis and the hookworm infection. It will see albendazole and pyrantel type of drugs we can give. If patient is having flu infection, then they can be treated with the paracetamol type of drugs. Then antiviral and antifungal drugs is there, and these drugs is effective on the viral infection and fungal infection. So the treatment of these type of drugs when the patient is suffering by a viral infection and fungal infection. Difficulty in treatment with the viral infection is that the virus is living and multiplying within the cell, and when they are multiplying, they will infect nearby the healthy cells. So drugs which enter the cell and not only destroy the virus but exert bad effect on the cells, also that we can utilize as an antiviral drugs. So antiviral drugs, many antiviral drugs is available for the viruses, and they have no cell wall. Viruses, you will see they don't have any cell wall or plasma membrane. Just they are two type of viruses, enveloped and non-enveloped type of viruses. And enveloped type of viruses, inside they are having capsid, and inside the capsid either they have DNA or RNA. So the very basic type of infectious molecule, when they are infecting to any host, then only they will be in the live condition, or they will start they are hijacking the cellular machinery, and they will start replicating and transcribing their own molecules. So viruses containing envelope antigenic in nature, and viruses are obligate intracellular parasites. So outside the host, they are like a dead particle. That's why they have to cause the infection, and then only they will divide. So they do not have a metabolic machinery of their own, and they use they will hijack the host machinery, and then only they will synthesize their viral protein and enzymes. So antiviral drugs is also very useful against these type of viral infection, and many antiviral drugs are purine or pyrimidine analogs, and many antiviral drugs are prodrugs. They must be phosphorylated by viral or cellular enzymes in order to become activate. So antiviral drugs agent inhibit active replication, so the virus growth will be, so the viral growth resume. So main purpose of these antiviral drug, it will inhibit the replication, so the viral infection will not spread in the body. Now, mechanism of action. Now, key characteristics of the antiviral drugs. So these key characteristics of the drugs, it should be there, and these drugs should be enter into the cells, and then it will be affected on the virus. Then it will interfere with the viral nucleic acid synthesis and regulation. And some drugs may interfere with the ability of virus to bind to the cell. Some drugs also stimulate the body's immune system, and it will best responses to antiviral drugs are in patient with competent immune system. So a healthy immune system works synergistically with the drugs to eliminate or suppress viral activity. Now, use of antiviral medication. So antiviral drugs used to treat infection caused by the viruses, and for HIV treatment we have anti-HIV type of treatment. So antiretroviral drugs we can give to HIV patients, and this then herpes simplex virus HSV1 oral herpes. The HSV1 type of virus is infecting on the oral, and HSV2 type of virus is infecting on the genital. Then varicella zoster virus is there, and this varicella zoster virus is causing the chickenpox. Then classification, if we see the DNA polymerase inhibitor type of drugs is there. These type of drugs will inhibit the DNA polymerase so that the DNA polymerase will not help in the replication of viruses. And when replication of the virus DNA or RNA, it will not be there, so virus can't able to synthesize new type of viruses, and we can stop the viral infection. Then purine and nucleoside type of analogs is there. Cyclovir, ganciclovir, panis, cyclovir, valacyclovir, panciclovir, and cidofovir is the purine nucleoside analogs. And these are all analogs can able to inhibit the DNA polymerase, and so that replication of the DNA virus DNA will be stopped. Then pyrimidine nucleoside analogs also there, and in this category idoxuridine is there, and non-nucleoside type of antiviral drug like foscarnet is there, and they are all inhibiting the DNA polymerase. Then inhibitor of viral penetration or uncoating, amantadine and rimantadine is there. These two type of antiviral drug can able to function on the viral penetration and uncoating. Then mRNA synthesis inhibitor like ribipirine and formimixane is there. Then neuramidase inhibitor like zanamivir, oseltamivir is there. Then immunomodulator like immunoglobulin, interferon, palivisms, and imiquimod. Imiquimod is functioning as a immunomodulator. Next group of drugs is antifungal agents, and these agents is having effect on the fungal infection and superficial or systemic type. Also, we are calling antimycotic drugs and antifungal and drugs and antimycotic drugs, they are both same. So they are used to treat two type of fungal infection: superficial fungal infection, skin or mucous membrane. When the systemic function infection is there, the fungal can infect in the lungs and central nervous system. So these two type of fungal infection is there, and these two type of fungal infection we can get different type of. Fungi is also known as a mycosis, and it is very large and diverse group of microorganisms. It broken down into the yeast and molds. If yeast infection is there, and we know that yeast is a single cell fungi reproduced by budding and very useful organism in the baking and alcohol production industries. Mold is multicellular, characterized by the long branching filamentous called hyphae type of organism. So fungal infection we are dividing in four general types: cutaneous, subcutaneous, superficial, and systemic. Systemic can be life-threatening and usually occur in immunocompromised host system. Candida albicans is one type of yeast type of. Candida albicans is infection, and due to antibiotic therapy and the neoplastic or immunosuppressant, it may result in the overgrowth and systemic infection. The Candida albicans can able to cause the infection in the mouth. Oral candidiasis or thrush it will produce, and in the newborn infants and immunocompromised patients mainly they will target. Then vaginal candidiasis is also there, and these vaginal candidiasis is a type of yeast infection. Vaginal candidiasis is mainly cause infection in the pregnancy, diabetes mellitus, and oral contraceptive type of. So here is the different type of antifungal drugs. Then these antifungal drugs will be having different type of mechanism. First is the glucocytosine is there. It will interfere with the protein synthesis. Imidazoles is there. They will interfere with the ergosterol synthesis, clotrimazole, and ketoconazole, tetraconazole, and terbinafine is there. These are all imidazoles. Then triazoles here, they will interfere in the ergosterol synthesis as well, and intraconazole, fluoroconazole, voriconazole, fosconazole is the type of triazoles. Then polines is there. These polines is amphotericin B, both administered usually the systemic type of infection, and it binds to ergosterol in the cell wall, causing leakage of material. So amphotericin B is the toxic to the renal tubes, and necessitating is an ointment for oral candidiasis. Netamycin is topical and used for eye infection. Then glycophyllene is there. These glycophyllene bind to creatine and precursor, making cell resistant to fungal infection. Then candine is also there. It will inhibit the glucans in fungal cell wall. Then other type of antifungal drugs also there, like donaflate, and potassium iodide, sulfonamide, penicillin, and undecylenic acid also we can give as an antifungal drugs. Now some important references we will see for the study of chemotherapeutic agent. So Tara B. Shanbag, Smita Shinoy, Pharmacology Preparation Manual for Undergraduate is there, and it is in the Elsevier publication. Again, Herbert Ashton, Nancy Clarkson is the author of the Pharmacology. Jones and Bartlett publication published this book. Then Govinda Smith's Pharmacology at a Glance, and the publisher is Paras Medical. Now we will see some important MCQs. So first one is, which of the following is a drug that interfere with the process of DNA production in the virus that cause genital herpes? So option A is erythromycin, option B is vancomycin, option C is amantadine, option D is acyclovir. So any one right answer you have to choose. Then now the next question is, which one of the antibiotic is not used as a food preservative? So option A is pimarazine, option B is niacin, option C is tylosin, option D is beta-lactam antibiotic. So you have to select any one correct answer. And next question, the most selective antibiotics are those that interfere with the synthesis of option A is bacterial DNA, option B is bacterial RNA, option C is bacterial cell wall, option D is bacterial plasma membrane. Then again you have to select the correct answer. Then next question, cycloserine inhibitors is, so option A is alanine racemase, option B is D-alanine synthetase, option C is alanine, option D is amidase. So you have to again select any one correct answer. And the last question is, which antibiotic has a beta-lactam range? Then option A is cephalosporin, option B is penicillin, option C is tetracycline, and option D is streptomycin. Here we are concluding the agent and antimicrobial, antifungal, and various type of drugs. We will start new topic in the next class. Thank you for being with us.